You off close up showing you what I have here, and the one labeled is phenylalanine. Okay, so my name is Kira Carlton, and I'm doing prompt 3.2b on the Nirenberg, Lita, and Myra approaches to decoding the genetic code. So, in 1961, American biochemist Marshall Nirenberg began cracking the genetic code, which paved the way for interpreting sequences of the entire human genome. So, him and his postdoctoral fellow from Germany, Matthias, they looked at mRNA and protein synthesis, and they used a cell-free protein synthesis. So they started their experiments together, and they studied linear molecule DNA and RNA. So they were able to identify specific nucleotide triplets that corresponded with certain amino acids. So the DNA nucleotides that they used were adenine A, guanine G, cytosine G, thymine T, and RNA, the uracil replaces the thymine. So how this experiment went down is Nirenberg and Matthias chose E. coli bacteria as a source of cytoplasm for the cell-free protein synthesis. So what they did was they took 20 test tubes, and each mixture had all 20 amino acids in it that are needed for translation. So in each test tube, one amino acid was radioactively logged, as I can kind of show there. And so the first thing that he used was only the nucleotide uracil called poly-U. And so he synthesized an mRNA, which poly-U kind of acted like as like a messenger directing the protein synthesis. And so what that does is it instructed a chain of amino acids in the phenylalanine. And so so he used this nucleotide uracil as a poly-U and synthesized an mRNA molecule. So put these in all of them. And so the only triplet in the poly-U is in the mRNA is UUU, as you can see here. And so he discovered that this UUU might code for phenylalanine because when he added it to the cell-free arrangement, the polypeptides that were made only had the amino acid phenylalanine in it. Okay, so what this experiment proved is that messenger RNA transcribes genetic information from DNA, and then that process is the assembly of amino acids to complex proteins. And in 1964, Nirenberg and Philip later discovered the way to determine the sequence of letters in each triplet codons for amino acids. And in 1966, Nirenberg uncoded 64 RNA triplet codons for all amino acids.